Sickle cell is a genetic disease that causes anemia, heart failure, kidney failure, and extreme pain. It is one of many genetic diseases brought on by a single letter change in the DNA sequence, a point mutation. What if we could find and replace that one letter? Genetic engineering, precisely editing the DNA of living cells, enables scientists to do this and more. Today, CRISPR is a tool changing the face of genetic engineering. CRISPR was discovered as part of a system bacteria use to defend themselves against bacteriophages. Bacteriophages attack their host by injecting DNA segments, hijacking bacterial cells. Consequently, bacteria evolved proteins to recognize and cut specific bacteriophage DNA segments using stored CRISPR RNA as their guide. This system has been harnessed by scientists for use in genetic engineering. Using CRISPR, scientists have the capability to cut DNA at specific places by changing the RNA guide. The Cas complex contains the molecular scissors that make these cuts. The first component of the Cas complex is the Cas protein, which acts as a helicase, unwinding the DNA, and as a nuclease, cutting the DNA. The other component is a strand of RNA that acts as a template, controlling where the protein makes its cut. The complex binds to the DNA, and Cas unwinds the double helix, scanning the sequence, comparing it to the guide RNA. When the complementary DNA sequence is found, the two nuclease domains of Cas each make a cut, resulting in a double-strand break. Okay, so the targeted DNA has been cut. Now what happens? Cells will automatically attempt to repair the break by two different mechanisms. In the first repair process, the two ends are simply connected. This process is prone to errors, and bases frequently drop out or are added, disrupting the sequence and compromising the gene's function. This can help researchers understand that specific gene's function or disable harmful genes. In the other repair, cells use a homologous piece of DNA as a repair template to fix the break. Scientists can therefore introduce a piece of DNA that is homologous on both ends but edited in the middle. This new DNA, which can have base pairs rearranged, is inserted into the original DNA like a Trojan horse. Before CRISPR's discovery in 2013, entire proteins had to be engineered to target individual DNA sites. CRISPR's target can be changed just by editing the guide RNA. CRISPR can also be used as a delivery system, transporting enzymes that can edit single bases or regulate transcription of genes to specific locations on the genome. CRISPR is also already being used to develop new treatments for retroviruses like HIV, to create animal models of genetic disease, and to edit immune cells to help treat cancer. CRISPR's ability to precisely edit genes also brings hope. A clinical trial currently underway is already showing some success in treating sickle cell with CRISPR. Instead of a lifetime of treatments to help minimize pain, CRISPR could someday provide patients with a cure.